So, hello everyone, my name is Michel. Thank you very much for joining us today to listen to Professor Tom Fantini, who will talk to us about does the repurposing of diabetes drugs offer hope for Parkinson's patients, a topic that will be of interest to all of us. I would like to remind everyone that this session is for information and education purposes only. So if you are seeking medical advice, diagnosis, or treatment, you should really consult a medical professional. There will be time for questions at the end of the presentation. Please use the Q&A function at the bottom of your screen. Please do not use the chat function for questions. For those of you who don't know us, I will just say a few words about Mark Lambert and I. We met at a tutorial on nutrition and Parkinson's in summer 2020 and decided to stay in touch. We became actually quite very good friends. We named our small group No Silver Bullet to reflect our belief in the need for a holistic approach to managing our symptoms. And we've been hosting fascinating speakers every few weeks to that effect. We post the recordings of our sessions on YouTube, and we also have an Instagram page and a Twitter feed. Mark, would you please post the details of the Twitter feed, the Instagram page, and the YouTube channel on the chat?
Yep, I'll be done in a minute.
Thank you very much. But let's come back to today's topic. Professor Tom Fantini is Professor of Neurology in the Department of Clinical and Movement Neurosciences at UCL Institute of Neurology, and consultant neurologist at the National Hospital for Neurology and Neurosurgery at Queen Square, London. Professor Fantini is responsible for Parkinson's patients who are undergoing advanced treatments such as DBS, deep brain stimulation, and Duodopa. He's the chief investigator for a series of trials of exenatide, which puts him in a particularly privileged position to talk to us about does the repurposing of diabetes drugs offer hope for Parkinson's patients. Tom, we are very fortunate to have you today. The stage is yours.
Thank you very much indeed. Thank you, Michelle. Thank you, Mark, for the invitation. So I'll get my talk up. Is that projecting okay?
That's working perfectly fine.
Great. So it's my absolute pleasure to try and address this question. Does the repurposing of diabetes drug offer hope for Parkinson's patients? So I'm going to give a story, if you like, to describe the epidemiology. This is trying to explain what the understanding is of our relationship is between diabetes and Parkinson's, and furthermore, look at the epidemiology linking diabetes drugs and Parkinson's. I'm then going to introduce the concept of there being insulin resistance in the brain. So insulin resistance is what is the cause for type 2 diabetes when it's peripherally. And so I'm going to talk about this phenomenon being perhaps relevant to brain pathological processes. I'm going to talk about laboratory data that informs us on the use of GLP-1 receptor agonists. This is a class of diabetes drugs, and how these may be applicable to Parkinson's. I'm going to talk about the trial data that we've been particularly involved in trying these drugs in Parkinson's patients, and give a summary update of the current situation. So to start with the epidemiology. So this is looking at how we can understand disease by looking at populations. And of course, you can look in subgroups of people, or you can look in the group of interest. Parkinson's affects lots of different types of people, and we have an increasing understanding about the genetic risks for developing Parkinson's disease. And we have some insight into some of the environmental risks for Parkinson's disease. What's become fairly clear recently is that having type 2 diabetes is an irrefutable risk for developing Parkinson's. So this is a paper published by a colleague of mine, Tom Warner. So he looked in the hospital records of 2 million people with diabetes. And he compared them with a group of 4 million people without diabetes, and looked to see whether there was any increased risk of developing Parkinson's longitudinally. And if you just take people who developed diabetes at any age, then there's a clear elevated risk. The risk is 1.3, so a 30% increased risk of developing Parkinson's if you have diabetes. What's even more convincing is that the size of the risk of developing Parkinson's subsequently depends on how old you were when you develop diabetes. So if you're in your 20s, 30s, or early 40s, when you develop diabetes, you have a risk almost 4 times that of someone without diabetes. Whereas if you are older, that risk soon becomes less. So if you only get diabetes in your late 70s, you only have a very marginally increased risk of later getting Parkinson's. So this, you can interpret this as a dose response effect. The more years of diabetes you have, the bigger the risk of subsequently getting Parkinson's. Now, not only is diabetes a risk for getting Parkinson's, diabetes is also associated with getting more aggressive Parkinson's. So this is work done by one of my colleagues, Dylan Athauda. He took data from the Tracking Parkinson's cohort. So many people may be signed up to this. This is a cohort of 2,000 people with Parkinson's disease. The data have been collected over the past 10 years or more now. And this group was separated between 1,700 people who have Parkinson's and another 167 who have Parkinson's and type 2 diabetes. And of course, they were followed, in this data analysis is over the subsequent 6 years. And what Dylan found is that if you have type 2 diabetes and Parkinson's, then you have much more rapid progression to developing cognitive impairment. So here the red line represents those people who are free of any cognitive impairment if you have diabetes as well, whereas the blue line is those people free of cognitive impairment without diabetes. And on the right-hand side, this is the similar comparison, people who have no gait impairment, so that as the line goes down, there's an increasing number of people with gait impairment. And of course, you again see that patients with Parkinson's and diabetes are getting earlier gait problems than people without. So having diabetes is not only a risk for getting Parkinson's, it makes matters worse, both from a cognitive perspective and a gait and balance perspective. Now, one question is whether this is all about blood sugar. So it's very likely that having elevated blood sugar is a contributory factor for a neurodegenerative process. This little cartoon on the left, the green is the circulating blood glucose. If glucose crosses into the central nervous system and is taken up by a nerve cell, some of the blood glucose can be converted to what we call advanced glycation end products, AGEs, and this is mediated by something called methyl glyoxal. Now, in normal health, these AGEs and methyl glyoxal are of no consequence, but in levels of very high blood sugar, then you get a lot of these AGEs, and these can cause problems to nerve cell health. And it's been demonstrated that there can be some negative effect on this balance that exists between having healthy monomers of alpha-synuclein, this is the protein which causes toxicity in Parkinson's, and having pathological oligomers of alpha-synuclein or fibrils of alpha-synuclein. So having more of these AGEs, these advanced glycation end products resulting from chronic high blood sugar, shifts the balance towards the toxic forms of alpha-synuclein. So this may well represent part of the reason why diabetes is bad for nerve cells and bad for the rate of progression of Parkinson's. Now, before qualifying that statement, I'm going to introduce you to the THIN database. So we learn the most reliable lessons from having very, very large data sets. It's all very well looking at small studies in tens or hundreds of people, but if you can look to see how illnesses and diseases affect thousands or millions of people, you can be much more confident about your results. So in the THIN database, this is over 100 GP practices in the UK, and it represents more than 16 million people who have signed up to have their health records indexed and searchable according to different illnesses and diseases. So within the THIN database, there's 13.3 million people who don't have diabetes, which gives us a risk of developing Parkinson's of 3.8 per 1,000 people. Now, in the diabetes subjects, of which there's more than 500,000 people in this database, the risk of Parkinson's is 10.7 per 1,000 people. If you only take those people who are not on treatment for their diabetes, and so these are the people with the uncontrolled high blood sugar, the risk is 13.9 per 1,000 people. So treating diabetes is better than not treating it in terms of your Parkinson's risk, but even if it's treated, you still have an elevated risk above and beyond people that don't have diabetes. Now, what we've been able to do is to look to see whether the risk of getting Parkinson's varies according to which drugs you're given. So here, this is a subgroup of the bigger data set where we have been able to identify people with new diagnoses of diabetes. So every single medication that they've ever been treated with can be accounted for in the subsequent longitudinal analysis. And you can divide people with diabetes into whether they have what we call control medications, that's metformin or sulfonylureas such as gliclazide and glipizide. So these are the most common drugs that are used to treat diabetes. And then there's a smaller subgroup that are treated with what we call the glitazones, here GTZ. And another group of people treated with DPP4 inhibitors, and the last group of people treated with the GLP-1 receptor agonists. And if you follow these arrows down, you see that in the control medication group, there are 10 cases of Parkinson's disease for every 10,000 person years of follow-up. It's slightly reduced in the glitazone group, where there's 8 cases of Parkinson's for every 10,000 person years of follow-up. It's, however, much lower in the group treated with DPP4 inhibitors, so 5 cases for 10,000 years. That's half of the rate in the control group. And it's lower still in the group treated with GLP-1 receptor agonists. Now, this is done with the most rigorous statistics, using what we call propensity scores, to make sure that these are comparable groups and there's no confounding that influences why some people were treated with the different classes of drugs. So the next thing I want to talk about is this phenomenon of insulin resistance. So as I said, type 2 diabetes is all about having insulin resistance in the body. You know, we have to contrast that with type 1 diabetes, where you don't make enough insulin at all, and so you're insulin short for that reason. But in type 2 diabetes, you make enough insulin, but your cells don't respond to it, and that leads to high circulating blood sugar. Now, the concept of having insulin resistance in Parkinson's and in the brain is relatively new. But this is work done by Vasilios Meissner, and this builds on some work that's been done in the Alzheimer's field for the last 5 or 10 years, where they've demonstrated insulin resistance in the brains of Alzheimer's patients. So Vasilios here has looked for insulin resistance in the post-mortem pathology, in the brains of patients with Parkinson's disease. And the way you can detect insulin resistance is by looking at if you like, the insulin receptor. It's actually insulin receptor substrate 1, IRS1. It can be phosphorylated to make your cell more sensitive, or it can be phosphorylated to make it more resistant. And here you see that there are fewer nerve cells. That's what we see with Parkinson's disease, but each nerve cell has a higher intensity for this insulin resistant phosphorylation of IRS1. So this is clear demonstration that insulin resistance can occur. We don't know if this is, causal, if this is the reason why cells degenerate, or if it's part of the phenomenon associated with inflammation or mitochondria not working or the proteins aggregating. But it's clear that this phenomenon of insulin resistance does occur in Parkinson's. So I'm now going to move on to talk about these drugs, the GLP-1 receptor agonists. These are the drugs that led to the lowest risk of our diabetes patients subsequently developing Parkinson's. So GLP-1 is glucagon-like peptide 1. This is a hormone that is released by the cells of the small and the large bowel every time you eat a meal. And it's released from the cells in the gut into the bloodstream. it circulates in the blood and it hits the GLP-1 receptors in the pancreas. And by hitting those receptors, it stimulates insulin release and suppresses glucagon release. Now, it only circulates for a few minutes before it's gobbled up by this enzyme called DPP4. And by stimulating insulin and suppressing glucagon at the time of eating a meal, it makes sure that your body doesn't get a big surge of blood sugar and keeps the sugar under control. Now, GLP-1 disappears within a few minutes, and that's because of this enzyme DPP4. But there are agonists for the GLP-1 receptor that don't get broken down by DPP4. These GLP-1 receptor agonists have a long-lasting effect on the pancreatic cells, and they've been now licensed for the treatment of type 2 diabetes because they enable a long-term control in blood sugar levels. There are also drugs that inhibit the DPP4 enzyme and enable your natural GLP-1 to circulate for longer, therefore having the same lasting effect on the pancreatic cells. So these drugs, GLP-1 receptor agonists and DPP4 inhibitors, are commonly used in patients with diabetes to control blood sugar. Now, if you look in the picture here, I've told you about GLP-1 receptors on the pancreas and what they do to insulin and glucagon. Interestingly, they also are good at keeping your pancreatic cells healthy by preventing them breaking down. So there seems to be a trophic effect on the pancreatic cells as well. There are also GLP-1 receptors around other tissues in the body. And the GLP-1 receptor agonists have been in trials and thought to be very beneficial at promoting liver cell health. They're also been studied in protecting heart cells in the context of a myocardial infarct. And of course, we're interested in what they do when the GLP-1 receptors in the brain are stimulated with respect to whether these are protective against toxins. So it's not just neurologists that are interested in GLP-1 receptors or diabetes doctors. These are of keen interest across different specialties. So the GLP-1 receptor agonists are this list of 6 drugs here. The first of them was exenatide, and then there have been some others launched more recently. They're all given by injection, either daily or weekly. The exception is semaglutide, which has recently launched an oral version of semaglutide, which can enable GLP-1 receptor stimulation without having to have the injection performed. So all of these are of potential interest, perhaps less so the dulaglutide and albiglutide, because those have been particularly engineered to have a big albumin attached to them, which means that they have absolutely no chance of getting anywhere near the brain because of the blood-brain barrier. But the other drugs are perhaps of great interest to us. So I'm going to tell you a bit about the laboratory data of how we or other people and some of my colleagues have been studying the GLP-1 receptors in the lab. To start with is the in vitro Parkinson's models. So this is work from 2002. So this story is now 20 years old. So you can poison nerve cells in a dish in the laboratory in a whole range of different ways. And one of the earliest and most accepted way of causing stress to a nerve cell is to expose it to a chemical called glutamate. It's actually a transmitter that our brains use all the time. But if you get exposed to too much glutamate, it's toxic to cells. And you should see here in the little graph that glutamate causes cells to die, but in the presence of GLP-1 or in the presence of exenatide, this toxicity is almost completely prevented. Now, more recently, their colleagues have discovered a little bit more about how exenatide might be helping nerve cells. What they've done is they've grown microglia in a dish. These are the pink cells here. You can think of these as being the policemen in the brain. You know, if there are any unwelcome pathogens or invaders or anything that's unwanted needs to be got rid of, the microglia will get rid of it. So what they've done in this experiment here, they've added these red preformed fibrils of alpha-synuclein to this culture of microglia, and the microglia become activated. And on the bottom graphs, you see they produce these toxic chemicals, TNF-alpha, interleukin-1A, C1q. And by releasing these chemicals, these are a way that these cells get rid of unwanted pathogens. And what they did in this experiment, they took the media, this is what the microglia are growing in, that contains these elevated levels of these toxic chemicals, and they added it to cultures of astrocytes. So astrocytes are the support cells in the nervous system. So you can think of them as your social workers. So if they're doing a good job, they're very helpful for your nerve cells. But if they're not doing a good job, then your nerve cells, don't get what they need. So in the presence of the media from the microglia, the astrocytes stop doing what they need to be doing, and they don't produce the right chemicals, and they actually become converted to a different type of astrocyte. And if you take the astrocyte conditioned media and add it to the nerve cells die. Now, if you add right at the start a form of exenatide that here is called Neuraly, then you stop the TNF-alpha being produced by the microglia, you stop the interleukin-1A, you stop the C1q, and it means your microglia don't lead to toxicity of the astrocytes, they're not converted, the neurons stay healthy, and no neuronal death occurs. So there's now a lot of knowledge about this way through which exenatide stops these activated microglia engaging in this unwanted inflammatory response. Now, doing experiments in dishes is all very interesting, and you can learn a lot, but what you want to do is to understand how these drugs affect normal, healthy mammalian biology. And of course, there's been lots of work done in mice and rats in different models that have been used to model Parkinson's disease. And this was done at UCL almost 10 or 15 years ago now. And you see here that in this 6-hydroxydopamine animal, the black bar is abnormal behavior, that these animals rotate in the presence of this 6-hydroxydopamine toxin. They spin round and round. But when you add exenatide in the green and in the blue in different doses, then you can stop this rotation. And in parallel with this, when the 6-hydroxydopamine obliterates the striatal dopamine levels, and in the presence of exenatide, this is reversed. And Peter Witten did this in 2 different animal models, the 6-hydroxydopamine and lipopolysaccharide, and found the same thing. What's been done even more recently is to look at the effects of exenatide in a different type of mouse model. These are mice that have been bred to express the human alpha-synuclein transgene. So mice don't normally develop Parkinson's, but if you add the alpha-synuclein transgene, then they develop alpha-synuclein pathology. And here, these mice have been allowed to grow for 6 months, and then they are treated with this exenatide for a 4-month period. And you see here at the bottom that what normally happens is that a mouse that doesn't have this abnormal gene will live for for up to 2 years. But in the presence of this abnormal transgene, the human alpha-synuclein, they start dying between the ages of 10 and 15 months. In the presence of exenatide, then you, by and large, you extend the lifespan by about 5 to 6 months. And the middle bars show the pathology. So in the top here, if you see my arrow, this is all the alpha-synuclein pathology that you see in these transgenic animals, which is halved in the presence of the exenatide. You see it quantitatively comparing the red and the blue arrows here. So in the most sophisticated animal models we have to try and recapitulate what is happening in people with Parkinson's, it's clear that these drugs have engagement, they can prolong survival and reduce pathology. So I'm now going to tell you about the trial data that we have using these drugs in people with Parkinson's disease. To start with, this is back in 2010. We wanted to see what happened using this drug compared to using placebo. we asked the current manufacturer of the drug at the time whether they would kindly give us some placebo, and unfortunately they said no. And the reason they said no was largely because of the cost of manufacturing a placebo and their fear that this was going to be a waste of their money and that the drug wouldn't work and wouldn't help people with Parkinson's. So faced with that, we decided to press on anyway, and we decided to do an open label trial when we recruited 44 people with Parkinson's, and we randomly allocated them to either have their regular Parkinson's medication alone, or they have their regular Parkinson's medication and use exenatide. And to make it as objective as possible, we record them at each visit over the next 24-month period. They use the injections for 12 months, but we had even longer term video follow-up. And we asked people who didn't know the patients to make a judgment, to score whether they were better or worse, in order to make a judgment about whether the drug had done anything. And this is what we found. This is the Movement Disorder Society UPDRS Part 3. So this is the standard, slightly old-fashioned way of scoring Parkinson's, looking at finger taps, hand taps, arm movements, walking, speech, balance, these types of things. And you see that over the course of the 12 months of using the drug, patients on exenatide had a clear advantage compared with people who were just in the control arm. But this advantage persisted even a year later, having stopped the medication. And likewise, when we measured people's cognitive performance, this is using the Mattis Dementia Rating Scale. In this group, for this scale, the higher the score, the better you are. The maximum you can score is 144. So people on exenatide improved on this scale, whereas people on the control arm deteriorated over the course of the 12 months. And things were pretty level over the subsequent year as well. So we published this paper in 2013, reporting these results. And of course, the biggest criticism is, is this a placebo effect? The patients knew that they were on the active drug, or they knew that they were in the control arm. And is this purely explicable by a placebo effect? But the major impact that this paper had was to convince the pharmaceutical manufacturer that the risk of getting involved with this research was less, having seen some data that perhaps we might be influencing the severity of Parkinson's disease. So we managed to convince Bristol Myers Squibb to give us drug and placebo. And in that context, we managed to convince the Michael J. Fox Foundation to give us funding to set up a double-blind placebo-controlled trial. And we designed a small trial. We could only get enough drug and placebo for 60 people. That was limited by the drug company. They gave us enough drug for 60 people for 48 weeks using once weekly 2 milligram injections. And we designed it so we'd have a 12-week washout period at the end, so we could see if any effects that are observed during the treatment are lasting or if they disappear. The patients had a disease duration of 6 years, and were using about 800 milligrams of levodopa equivalent during the day. And we used the same outcome measure as we had in the previous trial because it had been seen to be effective. And this is what we found. Patients on placebo, rather than having a placebo effect, which was kind of what we expected, they had a gradual but steady deterioration over the course of the 48 weeks that continued even through the washout period. So overall, they had a 2 points on the UPDRS Part 3 decline over this period of study. Whereas people on exenatide, they had a 2.5 point improvement really early on, right in the first 12 weeks. And then their scores stayed fairly static over the course of the treatment period out to 48 weeks. And then there was decline during the washout period of about 1.5 points. But what you can note is that the people on exenatide were still better off than they were at baseline, even at a time when the drug was no longer detectable in their blood. And they certainly didn't catch up anywhere near the rate of deterioration that the placebo group had reached. So we published this, and there were some criticisms from colleagues, and quite right, all science needs to be robustly critiqued. And one of the criticism was it's only 60 people. How can you possibly make a judgment on such a small sample? Another criticism was, well, perhaps because of the small sample, there were some slight differences in the characteristics of the patient at the start, and that may also influence their subsequent trajectory. Because exenatide does have side effects, it can cause nausea, it can cause weight loss, it can cause upset, either diarrhea or constipation. Perhaps those side effects unblinded people as to which arm they were on, and therefore that led to an additional placebo effect in the exenatide treatment group. These things are possibilities. Or perhaps the amount of levodopa people were using was adjusted by their doctors, or also maybe there was a bigger effect of the levodopa they were using because they'd lost some weight as a result of the exenatide. So all of these things were concerns. I think many people felt that there was something in this, that this trial did show that the drug was helpful, but there were nevertheless concerns whether this is just a symptomatic effect. And had we continued following people for more than the next 12 weeks, maybe those effects would have disappeared. So within this trial, we also took blood samples from people, and we wanted to study those blood samples carefully to see what extra we could learn. Now, nerve cells are constantly producing little vesicles, which are called exosomes. Most of the tissues of the body produce these exosomes, and these exosomes circulate in the bloodstream, and you can collect them from the bloodstream, and you can separate exosomes that have come from the central nervous system because they express this little protein on the surface called L1-CAM. So if you collect these exosomes, purify them for ones that have come from the central nervous system, you can then analyze their contents and see what's changed. And what we've found is that patients on placebo had little change, patients on exenatide had a big change. And what they changed is this protein that indicates insulin sensitivity. And back to our hypothesis about insulin resistance and insulin sensitivity in the brain having a role to play in neurodegeneration, this was entirely in keeping with this. And we also looked in these exosomes for a whole range of different proteins, a protein called AKT and a protein called mTOR. And both of these proteins are involved in brain cell survival. And all of these results are in keeping that our drug has engaged in this survival pathway due to improving insulin sensitivity. We did DAT scan imaging on these patients as well. And here you see that the placebo group, the yellow blobs are the deterioration in DAT scan uptake during the course of 1 year. The second row is the deterioration in the exenatide. So both groups have deteriorated. But if you subtract one from the other, there are some little yellow areas in the bottom row, which are the differences where there's an advantage in exenatide compared with placebo. And the bottom graphs show that these are small, but statistically significant changes seen. So to bring you up to date, we're now doing the third exenatide trial, and this is now a phase 3 trial. And what we want to firstly do is to see if we can reproduce what we found in the last trial in a larger number of people using multiple centers to recruit patients. So we're hoping that we see a small deterioration in placebo group, as we'd expect, and an advantage in exenatide. But the difference this time round is we're going to follow people not for 48 weeks, but they're going to stay either using exenatide or placebo for 96 weeks. And if we show that the exenatide group still flatline, the placebo group continue to deteriorate, that means there'll be divergence between the two groups' trajectories. And that divergence will be convincing evidence that this is not just hiding symptoms, that there's a cumulative benefit the more years you're exposed to the drug. And along the way, we're collecting DAT scans, we're doing genetics, we're collecting spinal fluid, we're testing the utility of smartphones to quantify tremor, slowness, and gait, or even using artificial intelligence software to get better ways of scoring hand movements. We're doing a small trial of using exenatide in patients with multiple system atrophy as well. This is a group that you'll be aware of have even more aggressive Parkinsonism and tend not to respond as well to levodopa. So we're seeing if exenatide might be helpful for these patients as well. There are, of course, trials of other types of diabetes medications. Back to the epidemiology at the start, you might remember there was a small advantage in using the glitazones in the epidemiology. So pioglitazone, which is the most commonly used one, was studied in this trial. This featured 200 people who are randomly allocated to have either one of two doses of pioglitazone or placebo, and they were followed for the best part of a year. Unfortunately, this trial didn't show any advantage, and the question remains: is it that the drug has too small an effect, or is it that you need to follow people for longer in order to see differences emerge? Insulin itself is of interest. Of course, you can't give insulin peripherally because it causes your blood sugar to drop, but you can deliver insulin nasally, and this may be taken up into the central nervous system and have effects on the central nervous system. And this has already been tried in Alzheimer's patients, and there's further interest in studying this in Alzheimer's disease with some positive data in the Alzheimer's group here. As yet, I'm not aware of anybody that's giving intranasal insulin in a proper controlled trial in Parkinson's patients, but of course this would be an interesting area to study. And lastly, there's a lot of interest in the GLP-1 agonists, not just exenatide. So the other form of exenatide, NLYL1, is the subject of a very large trial in the United States. And there's also trials of liraglutide, lixisenatide, semaglutide, and another form of exenatide in South Korea. So we'll hear more about the results of using these GLP-1 receptor agonists in Parkinson's patients in the next 12 to 24 months. So in answer to the question, does the repurposing of diabetes drug offer hope for Parkinson's patients? I hope very, very much that it does. I don't think any single drug will be a silver bullet for all patients with Parkinson's, or even for single patients with Parkinson's. What I think is more realistic is that a drug can reduce the rate of progression. And if it does reduce the rate of progression to a large enough extent, then that'll be great. But I expect that there'll be different drugs for different people and different combinations of drugs that ultimately will be identified to make Parkinson's disease a very, very manageable disease indeed. And that's what I hope. So I'd like to thank the patients that have contributed to all these trials and a lot of colleagues as well. So thanks for your attention.
Tom, thank you very much for this. This is very inspiring, very interesting. And the fact that you refer to the fact that there is no silver bullet echoes, of course, the name of our little group. We basically had Alberto Espe talking to us a few weeks ago, and he was basically very insistent on the fact that actually the patient himself is a puzzle, and that basically people have a bioassay which will vary from one person to the other. And I think that basically links up with what you said about GLP-1 potentially being a compound that can help some people or can be part of some people's array of weapons. Thank you very much. May I just ask you to stop sharing your screen, perhaps, if you don't mind? Thank you very much. So let's just discover the questions that we have so far. I can see we have 9 of them. Actually, the first question is from Audrey. And Audrey, as you can perhaps see if you click on Q&A, basically saying that GLP-1 agonists seem to increase insulin. So she is not sure it's a good thing, as insulin-resistant people already run high insulin, which is making them ill. Can you elaborate on the mechanisms that are at work, please?
So I think GLP-1 receptor agonists from a Parkinson's perspective are not going to have an impact on brain insulin. I think that the impact is on insulin sensitivity. So I don't worry that there's going to be excessive levels of insulin in the brain, which are going to be a problem. In diabetes patients where there is insulin resistance, and as a result of a GLP-1 receptor agonist, if there's chronic GLP-1 receptor stimulation, these drugs are helpful at boosting insulin release in a glucose dependent level way. So if you have a high blood sugar, then you get an insulin release. If your blood sugar is normal, then insulin is not released. So there's glucose level dependent insulin secretion, which means you won't get excessive insulin above and beyond what you have already. So that's really a question for the diabetes doctors, but as a Parkinson's doctor, I'm not concerned that GLP-1 receptor agonists are bad for people with diabetes. They've been licensed and used for decades. In Parkinson's disease, it's not that they're going to boost insulin levels to a pathological way. It's more their impact on insulin sensitivity in the brain that is how we hope they'll function.
Thank you very much. Another question from Audrey, who says that so if someone is lean, athletic, and fit, like you and I and her, I think, from what she said earlier, how will they benefit by taking exenatide, as they certainly don't want to lose appetite and weight?
So this is the disconnect between your peripheral metabolism and what's going on in the brain. We don't know how strong the link is between peripheral insulin resistance, which is associated with generally being overweight and sedentary, and central insulin resistance that we think is a contributor to Parkinson's and neurodegeneration. It might be that there are some people that have genetic risks for both. It might be that some people are saved from Parkinson's disease because they have elevated levels of circulating insulin in the absence of diabetes. So this is an interesting report that having an elevated body mass index but not having diabetes is actually protective for Parkinson's. So it might be that having higher levels of circulating insulin is actually good because that insulin gets into the brain. But as soon as you become insulin resistant, you actually become diabetic, then you pass that point of no return. And whether it's elevated blood sugar or whether it's a consequence of that causing insulin resistance in the brain, that needs to be elucidated.
Thank you very much. So sorry to our participants, I see that some people are still posting questions in the chat section. If you don't mind posting them in the Q&A, because I will not be able to pick them up from the chat. Martin Rumsby, I think you posted something in the chat. I think I don't want to lose this. So from one of our participants, someone is asking about Ozempic, and I don't know if you're familiar with that drug. He's asking whether that drug works in a similar way of exenatide because he's unable to purchase exenatide where he lives.
So Ozempic is another one of the GLP-1 receptor agonists. I think that's semaglutide. It shouldn't take me a second just to look that up. But yes, so I suspect that if one, yes, it's semaglutide. If one GLP-1 receptor agonist works, then the advantage between them might be because of central nervous system penetration. But if one works, hopefully they will all work. The question about semaglutide is whether it gets into the brain. There are some data that cast doubt on whether semaglutide gets into the brain. But I've spoken to the manufacturers of semaglutide, and they wonder if it's necessary. It might be that by having a peripheral effect, then that does influence whether it's cytotoxins or whatever else that do get into the brain subsequently. If you have a beneficial effect peripherally, if that can still have a beneficial effect centrally, even without the drug getting to the target. So the true answer to that question is, I can't recommend anybody use any of these drugs. We're still collecting the data to support the use of one drug, and then we'll have to wait for the trials that are being done on semaglutide to recommend Ozempic as well.
Perfect, that makes sense. Donna is asking whether the 44 people in the exenatide trial were tested from a genetic standpoint.
The answer for that is we didn't get genetic samples of everybody, and because there were so few, we only had a handful of people who were positive for any of the Parkinson's genes. So we didn't have the power to detect anything interesting from that.
And a follow-up question from Donna, still on the same 44 people who I understand were quite a small group, but during the trials, did the participants change the amount of Sinemet they were taking? And also, did they have the same classification of symptoms? Were they homogeneous?
So in terms of Sinemet or Madapar, so there was no restriction to changing levodopa, but when we did our analysis, we adjusted for their levodopa equivalent dose in the analysis. So if someone had changed, then that was part of this the statistical comparison. So that meant that, because it's not really ethical to say you have to be on the same levodopa regime for a whole year. You know, people on placebo, particularly if they are deteriorating, they need to be able to adjust the dose, and you have to have the same for both groups, otherwise it becomes unblinded. So you just have to adjust for this in the statistics, rather than place restriction on patients that end up dropping out as a result.
Okay, Jerry is asking, I don't know if you know the answer to that, but when do you expect the readout of the MSA study? I don't know if you have an idea when it could come out.
So we finished recruitment to the MSA in, well, it was the start of May, end of April, start of May. And so there's a 48-week period of drug exposure. So we'll have that data February, March in 23.
Thank you very much. So 2 questions on NLY-01.
Yeah.
Dan is basically saying that he is on the NLY-01 trial, and he's asking, how is this form of exenatide different from the version of the drug you have worked with?
So NLY-01 is pegylated. So pegylation is a PEG, polyethylene glycol, addition to the exenatide molecule. And that means it is thought to penetrate the blood-brain barrier to a greater extent. It's more lipid soluble. So it's really that delivery into the central nervous system, and perhaps also its half-life in circulation as well. So it's what it does to the receptor is exactly the same, but dose for dose, you may get longer and better penetration. You know, we don't know how much you need, and whether there's, you know, we're going to be limited by side effects or just insufficient dosing centrally. So, you know, we'll have to wait and see what that trial produces.
Thank you very much. Another question from someone else on the NLY-01 trial. That person has heard rumors that the trial in the US was going to have an open label extension, but that this was postponed indefinitely. Have you heard anything about that?
I don't, you know, I haven't been involved in the trial at all. I know one of the people that did a lot of the basic science prior to the trial, and so I know a lot about the drug, and it has been very robustly studied, but I don't know the trialists to have any insight.
No problem. Becky is asking if the phase 1 trial is still open.
So our current trial, now we've finished recruitment, and that was fairly recently. I think it was the 20-something of April, and that means that from 96 weeks from then we'll be finishing. We'll have the data from that.
So it is too late for Adrian, who is asking if he can take part in the trial, phase 3 trial.
I'm sorry. Yes, we've had to close recruitment, and the main reason we had to close by a certain date was because of drug expiry. We recruited people throughout the COVID pandemic. Of course, we had to stop recruitment during the lockdowns. But we got a succession of delays, and that meant that our drug expiry data got closer. So we had to think very carefully of that to decide when to complete recruitment.
Thank you. Stephen, who is a good friend and actually a GP, is asking if exenatide would have any use as a preventative drug for PD, and then when would one use it?
So interesting. So difficult question. Thanks, Stephen. Of course, we have to do one thing at a time. If we show it's helpful in Parkinson's disease, that's fantastic for the almost 10 million people around the world with Parkinson's, and we'll have to look carefully about if there are subgroups that it has a bigger effect, and perhaps subgroups that it doesn't have so much effect. If we found that it had a particular benefit for an easily definable subgroup, let's say LRRK2 mutation carriers or GBA mutation carriers, then of course those people can be identified in the prodromal phase before they have any symptoms of Parkinson's. And then it would be quite logical to say, well, if the drug helps those people and we can identify them before they've developed motor PD, then it's not such a big step to say, well, it could be used as a preventative therapy. But the trial would have to be done. There is a trial being set up by the Michael J. Fox Foundation looking for agents which prevent Parkinson's disease. And they're using people that are either gene positive carriers or people with REM sleep behavior disorder as the population at risk. And of course, there are challenges in doing this, but there will be platforms available to try and look for preventive agents, not just treatments as we're doing.
Thank you very much. Question from Chris, who is basically asking if the evidence of correlation between high blood sugar, unregulated diabetes, and PD suggests that a low sugar diet is worthy of exploration, perhaps something of a ketogenic nature.
So this is where we start speculating rather than dealing with facts. I would very much advocate a healthy diet and plenty of exercise for any patients with Parkinson's I see. We know from Bas Bloom's work that exercise in of itself can keep UPDRS scores low. And so, I think not just improving flexibility and core strength, but actually the metabolic benefits of exercise are really important. Having diabetes is very bad for you, but what the population who don't have diabetes, if they can influence their risk for Parkinson's by one dietary form or another, I think it would be better just to make sure you have a diet full of antioxidants. Fruit and vegetables, these types of things are far more likely to be helpful than trying too closely to manipulate blood sugar.
Bas Bloom, by the way, will be a speaker here in September, so that will be an interesting topic too.
You'll enjoy every minute of Bas.
David has a question in the same kind of direction as the one I just put to you. He's basically saying, is it too much of a left field query to think that reducing sugar intake as people age would help offset the risk of developing PD as well as diabetes? Similarly, he's asking that if the increased level of risk of obesity through the UK means there is a real problem with people in the future developing PD as well as diabetes and other related illnesses.
So I don't know. What's thrown the sort of the cat among the pigeons is this observation that an elevated BMI in the absence of diabetes is actually reduces the risk for Parkinson's. So that might be slightly confounded, and it's just thin people are at higher risk of Parkinson's for whatever reason, and people that are, let's say, normal to slightly overweight, that's the baseline, and then people with diabetes are at elevated risk again. So it could be an inverted U-shaped curve that there's an optimal. But as I say, I don't think we know enough to advise trying to manipulate blood sugars in the healthy range. Of course, you want to avoid diabetes, and you want to make sure that you have a good exposure to antioxidants. Thank you, Tom.
So let's go for an easy question before we go for a highly technical one. So the easier one is basically, would you be prepared to put a time scale? Well, easy to tell us, maybe you don't know, but could you put a time scale on the availability of exenatide as a drug for PD patients if it confirms its initial results?
Okay, so if we have positive data, then generally you need 2 phase 3 trials showing the drug works in order to convince regulatory authorities to license it. But they will license a drug on the basis of 1 phase 3 trial if the effect size is big enough. If it's clear that patients on exenatide are doing so much better than patients on placebo, then that will be enough to convince the regulatory authorities. What the process to get a marketing authorization, it's possible without a pharmaceutical company, but it's much easier with a pharmaceutical company. So they have to produce all the data about the safety, the toxicology that's been done in animal experiments prior to all the diabetes launch. And with those data, they can then submit the efficacy data in Parkinson's and apply for a license. Now that process depends on the regulator's timescale, but I would expect about a year.
Maybe just a question from my own here, actually, on that topic of marketing and support by the pharmaceutical company. Is the pharmaceutical company behind exenatide excited about the potential results and the potential use for PD? Do they view this as a big opportunity for them?
So AstraZeneca are making exenatide for diabetes. At the moment, they don't have a big neuroscience interest. And so we will have to drag them to applying for this marketing authorization and point out that this is in their commercial interests. It may be that Neurally, who are trialing this pegylated form of exenatide, or Peptron, who have got another modified form of exenatide, it might be that if they publish their results in a similar time frame, that we have 3 trials that if they're all consistent, that it might be that some other big pharmaceutical company decides to buy Neurally and then launch Neurally with all the marketing to actually make a product, or to buy Peptron, or to buy the rights from AstraZeneca. And I suspect it's just a strategic decision that these big pharmaceutical companies make. And it's often easier for them to do so rather than a small company like Neurally or Peptron to venture into a global market.
Thank you, Tom. So as I said, I have a technical question. There are always questions, not being a scientist, which I will tell you frankly, I do not understand very well. So this is a question from Martin, who I know very well, and he's a chemist and a professor, a university professor. He's basically saying capillaries in the CNS are not leaky due to tight junctions. So how do exosomes get from the CNS into the bloodstream in the results that you showed?
I don't know, is my simple question. I know you can, when you purify the exosomes, you size them, and you can be confident that they are exosomes, vesicles, but according to their sizing. And you can also measure proteins within them that are exclusively produced by the central nervous system. So tau, for example. So if you show that the exosomes that you're harvesting and purifying with your L1-CAM antibody, if they contain tau, then they must have come from the central nervous system, because that protein isn't produced anywhere else. So I can't answer his question because I'm too busy in clinic and doing trials, but I'm sure there are scientists that would give him a better answer.
Thank you, Tom. That's already much better than any of us could have given you. A question from Ketan, who is basically asking whether there are any trials being considered with DPP-4I drugs, as it seems they have a similar lower risk percentage in developing PD. And he's also saying, should I discuss switching to DPP-4I from metformin with my GP?
Yes, so there's a trial looking at, I think it's alogliptin, which is one of the DPP-4 inhibitors. That's going to be run in a multi-arm trial in Australia. So I don't know the start and end dates. I suspect they're already recruiting to that. The advantage is that it's oral, so it's less inconvenient than having to do the injections. Of course, the DPP-4 inhibitors don't get into the brain, but they don't have to, because they're having an effect on GLP-1, and if sufficient GLP-1 gets into the brain, then it that issue doesn't matter. So in terms of his personal question about switching from metformin to gliptin, it depends on how well his diabetes is controlled. And I would look at this in its whole. If he has If he has excellent diabetes control and he's modified any other risk factors, then I would wait for the trial before switching. You don't want to jeopardize your diabetes control by switching drugs.
Exactly. Thank you, Tom. Harry is asking whether basically he presumes that GLP agonists might lead to basically decrease in L-DOPA dosage or reduce stimulation parameters in patients receiving DBS. I know that you're also a DBS specialist and L-DOPA specialist. What do you think?
Well, maybe. If the effects that we saw in the last trial, of having a couple of points improvement in UPDRS scores from day one, basically, then if that's the case, then there's no reason why levodopa doses couldn't be reduced, and likewise DBS parameters reduced. But I suspect it will be a cumulative thing. You get additional benefits, and then staying on the same levodopa dose means you don't go back to how you were. And so, provided the levodopa is not causing, dyskinesia or DBS causing some other unwanted problem, then you might get the benefit without needing to change.
Thank you very much. Ken is basically saying that the liraglutide study seemed to have mixed results. What is your reaction to the data?
So, I mean, all I know is what Michele Tagliati has presented. I've not seen the raw data, and it's not been peer reviewed. It's a small trial, is the first thing to say. They had some benefits on non-motor symptoms, and they had some benefit on UPDRS Part 2, which is people's activities of daily living, but they didn't have any benefit on UPDRS Part 3, which is the clinician's objective rating of Parkinson's severity. I think the fact that there was a signal, that's encouraging, but we've had positive signals in 2 trials with exenatide, and I'm not yet overwhelmed that we have all the data we need to start prescribing the drug. I think we need to have the results of a larger phase 3 trial, which is what we're doing, and I will look at that in the context of all the other trials which we're reporting around the same time, and try and make the best recommendation for people with Parkinson's, as well as to getting the drug approved if that's what the data is supportive of.
Thank you, Tom. Dominic is asking whether exenatide could potentially reverse PD progression, or is he asking for too much?
Well, could it reverse? I suppose it depends, obviously, what we ultimately understand about how it works. If you think of Parkinson's being healthy dopaminergic nerve cells, or healthy nerve cells in general, it's not just dopamine, or dead nerve cells, then of course it may stop cells dying and stop things getting worse. In terms of reversing progression, if there's a middle ground where you can have healthy, you can have alive, but just quiescent, so the cells are not producing dopamine because they're sick, but they're not yet dead. If you give an intervention, whether it's exenatide or something else, and it allows that cell to function again, then it can start producing dopamine again. It can start actively functioning, and then of course that could mean that things can be, back to how they were. And so if that means reversing the development of symptoms, then yes, then I don't see why not.
Thank you. Randy is actually sharing his experience. He says, I have been taking exenatide for over a year, along with other people with young onset PD patients, off-label. Everyone is fit. Everyone has seen improvements and slowing of progression. No one has dropped off. And because of that, all have reduced Sinemet use. So he says, thank you for your persistence. Sorry. He also has a question. He says, does Bydureon exenatide have an impact on noradrenaline levels?
Well, I mean, firstly, it's great to hear his feedback, and the more people that tell me that exenatide is helpful, that the more reassured I am that we're onto something and that we're not causing people harm. If I worry if people tell me that they're using exenatide and they're getting side effects, because I don't know if it's doing them any good yet. Yeah. But if he's feeling well and doing well, then with good symptom control, then that's fantastic. Does it do anything to noradrenaline? Possibly. There are GLP-1 receptors in the brainstem, including in the glossopharyngeal and nucleus and area postrema, and so there is some adrenergic nerve cells there, but I don't think anybody has measured levels of noradrenaline in the blood to see if there's any effect. But of course, dopamine and noradrenaline are very closely linked neurotransmitters, so having an effect on one can theoretically have an effect on the other. Yes.
Thank you. Just a quick word for Keith, who asked the question. Keith, I'm sorry, can you please rephrase your question? I think that there are some bits that I don't understand. I will mark it as answer, and I will let you just...
I think you're saying, is it likely to be effective for people with PSP?
Yes. Oh, good. Well, if it makes sense to you, perfectly.
Yeah. Well, maybe. I mean, of course, PSP is a tauopathy. It's not an alpha-synucleinopathy. There are no published data about insulin resistance in PSP, but we all live off anecdotes as well. And in a patient that was a participant in a trial he thought to have Parkinson's disease, we subsequently discovered that they had PSP, and they had a very high level of when we did the assay for looking at exenatide in the central nervous system, they had a very high level, and I suspect that their anecdotal case report would be enough for us to want to pursue it further, but I don't have anything more scientific than that to say.
Thank you. I like the way Teresa is cutting the chase here. She says, I don't understand all the technical info, but in summary, should people with PD take exenatide? I think your answer will be that it's premature at this stage. Correct.
It is premature, and I have this conversation almost every day at the moment. That I would love exenatide to be proven to be effective. If someone has diabetes and Parkinson's, and they're asking me that question, then I think it's an easier way to hedge your bet, saying, well, yes, if you use it for your diabetes, provided you've got good diabetes control in general, then there's less risk. But if someone just has Parkinson's at the moment, up until recently, I've encouraged them to participate in the trial, and now we have to wait for those results.
Thank you very much, Tom. Someone is asking whether there is evidence that people with Parkinson's have an increased risk of developing type 2 diabetes.
There's no evidence for that at all, largely because Parkinson's is later age of onset, but people have looked at that and there hasn't been anything.
Thank you. Donna basically is referring to the discussion we had with Alberto Espay a few weeks ago. Alberto, as you know probably very well, is advocating that trials should take into account the specificities of each patient and not just the specificities of the drug that they're testing. Are you taking this on board, or are you sensitive to his what he's advocating?
Yeah. I mean, I agree with Alberto. I think exenatide, if it has an effect on inflammation, then it will have relevance to the majority of people with Parkinson's. And so we've been quite broad in our inclusion criteria. And if we have a positive effect, then we'll only have a positive effect if there is a response by the majority of people. If only a few people respond and most people don't, then we won't see that. What we looked at after the last trial, we did a post hoc analysis, and we tried to identify whether there were any clinical features which were associated with response. And we found that younger people with tremor dominant disease, interestingly, who had more disability in their activities of daily living, those were the people that did the best. But it's not that older people or people without tremor didn't respond, that there was also some improvement across people. So I entirely agree with Alberto that there are subgroups of Parkinson's that need a precision medicine, and it might be for people with GBA Parkinson's disease, they need a lysosomal treatment. Some people with mitochondrial, so Parkin, FINK1 mutations, they need a mitochondrial treatment. But I'm still a little bit optimistic as well that there is a common contributing pathway of neuroinflammation, or things that are related to alpha-synuclein aggregation that will have beneficial effects for the majority of people with PD. And we don't need to be so stratified in our approach. So yeah, yes, but not everybody.
Excellent. Thank you very much. Stephen has another question. He's basically asking if proven as effective for treating PD, would exenatide be needed for life, or is it more temporary intake we should expect?
So the people that have been in the trials, many have wanted to stay on exenatide afterwards, because they've used the drug, they've felt there's been a benefit, objectively there's been a benefit, in our scores, and then when they've stopped the drug, they felt they've started to decline. Now, in our first trial, we followed people up after a year, and there'd been some stability, but even with that stability, when you keep seeing people year on year, Parkinson's again starts to deteriorate, and you get your arm twisted by people saying, look, I was doing well on this drug, how can you not give it to me now? And so I have this conversation with people, and if they're coming to my clinic under my care, then I'll have a different conversation than someone that just wants to approach me remotely, because I don't want to take responsibility for people who I don't know their safety, or for a drug that isn't proven to be helpful. But in someone that we have data showing that they have a good response, they tolerate the drug, then I try and find a way with their GP to try and to continue that prescription.
Thank you very much. Chris is basically touching on the question we spoke about a tiny bit before, but without being very specific. What would be the earliest timetable, do you think, if successful, phase 3 trial for the availability of the drug for PD patients?
Yeah, so I think it is likely to be at least a year after we have our data that I think it would take that long to go through even the accelerated licensing process, and then even when, let's say, the EMEA or FDA or MHRA, whoever decides that the threshold has been met, then in the UK we'll be at the mercy of NHS England and NICE to want to know about cost effectiveness, and be convinced that there's a health economic reason to allow this drug to be prescribed. But of course, we can shout loudly and use all the charities and platforms that we have to try and make that argument. Thank
you very much. Audrey and Peter are both touching on keto. Basically, Audrey is saying, what about giving the brain an alternative fuel source like ketones? And Peter apologizes for joining a bit later, and basically saying, has metformin been discussed? Is metformin seen as an effective drug in PD as an addition to my ketogenic diet? So I don't think glucose as a fuel source is the problem. So I wouldn't say that using ketones instead is another way. I mean, there may well be a role for ketogenic diets in Parkinson's disease, but I don't think that has any relevance on the role of the GLP-1 receptor agonists in Parkinson's disease. In terms of metformin, there has been some laboratory data to suggest metformin might be helpful, but in our epidemiology, where we looked at glitazones, DPP-4s, and GLP-1s, those were compared against metformin and sulfonylureas, and there's a clear advantage in the other group. So I'd be less inclined to explore metformin. Thank you, Tom. Someone is asking about the DBS patients and whether they would benefit from exenatide in the same way as other people might. There's no reason why if we show that the drug is helpful for the broad population with Parkinson's, DBS patients are no different. So there's no reason to think not. Thank you. Craig is asking about what has been found in PD3, and if the benefits are both neuroprotective and neuroregenerative. So we
we've finished recruitment, we're following people up, and we'll have data, about 18 months from now. So we, until it's unblinded and we have a look at the data, we can't say.
A bit too early, yes. Adrian is asking whether there is anything preventing PD patients from using exenatide. Now, I guess it's the same question we've been touching on before. Before it is a proven drug, they should not be taking exenatide.
Yeah.
Unless they have diabetes.
Yes.
So Adrian has another question. Does the data obtained so far give any indication that there may also be a restorative improvement to issues with gait and also freezing from using exenatide? I think we have seen some impact on gait in the use of GLP-1, as you were mentioning before.
So I suppose when we see gait freezing and balance problems, those tend to herald involvement of non-dopaminergic pathways in Parkinson's. It tends to be the cholinergic nuclei in the brainstem and the base of the brain that are involved. I don't know that exenatide won't be helpful. We had some benefits of cognition in the first trial. I suppose that the question is whether those are reversible, and that goes back to my answer previously about things might only be reversed if cells haven't degenerated, that they're just dormant, and if you can salvage some of their machinery, then they can start functioning again. So we have to wait and see.
Very good and practical question from Donna is, how expensive is exenatide?
So it's about £100 a month. You can shop around in different pharmacies, and you can some pharmacies will be charging double that. So if anybody is using it for whatever reasons, then do make sure that the price you're paying is competitive, because not all pharmacies charge the same.
Thank you very much. Are there any published bad side effects for exenatide based on basically people having type 2 diabetes using it?
So the common things are nausea, tummy upset, and so it's with the nausea you can lose your appetite, and it's that combination of nausea and loss of appetite which can lead to weight loss, and then tummy upset, diarrhea, and constipation. These things are common. They tend to disappear within the first 8 to 12 weeks of using the injections. So when you first start using injections, you get spikes of exenatide, then it falls down, then a spike and up and down. But after the first 2 to 3 months of using it, then the levels become more stable, and that means that lots of these side effects disappear. The only thing that doesn't stop is the weight loss. So people can lose about 3 kilograms per year. That was our experience. And exenatide is licensed as a treatment for people with obesity. So this is a well-recognized side effect. If you look at the patient literature that comes with the exenatide drug, it's listed 2 things which are worrying. One is pancreatitis, and one is thyroid cancer. Now, in the manufacturer does monitoring of all, surveillance of all the diabetes patients on exenatide and related drugs. And when they issue reports annually on this, and in the latest reports, there is not considered to be any elevated risk of pancreatitis, nor any elevated risk of thyroid cancer. So these are theoretical things that were reported when the drugs were first launched, but in the biggest meta-analyses and in the latest reports, that they're not thought to be a credible increased risk.
Thank you, Tom. I'm just revisiting something you touched on before. Someone is asking here, should the phase 3 trial be successful, do you think you could get a regulatory approval with a single phase 3 trial? Or do you think you would need 2?
So generally, the regulatory authorities say they like 2 trials, but they're prepared to make a license on the basis of 1 if the data are sufficiently convincing. So both statistically, you need a good p-value, but also an effect size that that's clinically relevant. So it is possible, but we have to see.
Okay. Just a comment from Robert. He says he's been on Vidureon for 2.5 years, off-label, no side effects, and my doctor tells me I'm stable if not improved. So stable for us is already something. And then the last question is from Dan. It's a nice way to end this presentation and Q&A session. He's asking whether you plan on attending the Grand Challenges in Parkinson's disease, modifying progression in Grand Rapids in this fall at the Vandal Institute.
There's a meeting that's back to back with that called the International Link Clinical Trials meeting, which I will be attending. So I will be in Grand Rapids, but I think the ILCT meeting finishes, as the Grand Challenges starts, and I'll probably be coming back to the UK, within a day or so of the Grand Challenges starting. So I won't be there for much of it, I'm afraid.
So Tom, thank you very much. And thank you to our audience for so many questions. I can't remember exactly how many we had, but probably like 45 or 50, which demonstrate a very strong interest. Tom, thank you for your time. You've been very generous with our audience and with us. I'm sure that not only we had the live audience today, but we'll have thousands of people watching you on YouTube and being very interested in your presentation. Thank you so much.
Thank you very much indeed. All the very best. Thanks for the invitation.
All the best. Bye-bye.
Bye.